Today we have an alumna coming back to present on her work in graduate school in conjunction with the Career Center, who helped coordinate this visit. Tendie is a 2010 graduate of CMU, and she has some family ties to CMU as well, where she's had a grandfather and an aunt. Is that who?
Yes.
Yes, who also have graduated from. After graduating from CMU with a degree in math, Tendie went on to start her work on her PhD in biomedical engineering, and I think she will discuss some of the issues or things that has arisen in that discussion as well, and she made some fairly bold decisions in regard to her education as well, some of the choices of moving while in graduate school to a different school all together. From Zimbabwe, I'd like to present Tendie Muzorewa and her talk on her experience in graduate school. So please join me in welcoming Tendie.
Thank you very much for the introduction, Dr. Belfay. As Dr. Belfay mentioned, I'm Tendie Muzorewa. Thank you so much for coming, those of you who had a choice. My topic today is mathematically speaking. So a lot of you people in this room are up here on this graph of how much you know about the topic you're studying versus what you know about it. So you've probably taken introductory biology or general chemistry, and you know a little bit about a lot of stuff, but you haven't really gone in depth in any one topic. I, on the other hand, am somewhere over there on this graph and making my way to PhD, and I'm hoping to convince you by the end of this talk to join me down this slope. So I'm going to tell you a little bit about myself and then tell you why you might consider graduate school and give my experience as an example of what graduate school is like. So as Dr. Belfay mentioned, I was a math major here. I graduated in 2010. I was a physics minor. There weren't any physics majors back then, so I didn't want to be the only one. I took a lot of biology, chemistry, and I also took some computer science courses. Now I mention this because in graduate school you have the option to design your own curriculum more or less. And when I was here as a math major, I always knew I was going to go into biomedical engineering. So I specifically chose these courses knowing what I was going to do. So I'm in my third year at Drexel. I'm a PhD candidate, which means all I have to do is write my dissertation and then they'll give me the PhD. It sounds a lot easier than it is. I'm in the cellular biomechanics lab, so that's where I'm doing my research. So why I chose graduate school? Firstly was for the challenge of it. Graduate school isn't like undergrad where they give you a PowerPoint presentation or however they teach you, take notes, and they give you homework and you can look up the answers on Google or in the textbook. The answers aren't online or in the textbook. You're asking a question that maybe nobody else has asked and you're trying to answer it using the information you have or the skills you've acquired. Also, like I mentioned, you have the freedom to study what you want. Of course, just like here, you're going to have general education courses, but you also have the option to pick the courses you want. There are a lot of specialized courses that you can take that are very specific to your research. And if where you attend graduate school, there are faculty who are also interested in this, then you have that advantage of really going in depth into a very specific topic. Right now, I'm taking a course in the mechanics of nanobiomaterials. So it's not materials, it's not nanomaterials, it's not biomechanics, it's a very specific course. And so that's very interesting because I don't have to take a whole bunch of courses I don't want to that won't really affect my life or help with my research. The next thing is that there are lots of rewards to graduate school. I mean, you get a sense of accomplishment, it's hard work, and when things are going well, they go really well, and you're like, oh my gosh, I'm going to win a Nobel Prize. And when things are not going so well, you're really down in the dumps, staring at your computer, pulling your hair out, but when it works, it's a very great feeling. And by the end of it, hopefully, you'll be the one person in the world who knows a lot of what there is to be known about your dissertation topic. So what can graduate school do for you? Well, there are a lot of statistics coming out suggesting that people who graduate from college from a 4-year program end up working in jobs that don't require a 4-year degree. So this pie chart based on information from the US Census Bureau shows that 62% of college graduates are working in a job that requires a college degree. That means about 40% of people are going to graduate and work at fast food places, even though they have a college degree. This has a lot to do with the economy. So you are looking for ways to set yourself apart from everybody else. There's also information that suggests that only 27.3% of people are working in a job that is related to their college major. There are a lot of math majors out there that then go out and work their way up in a construction company. They don't really need their math degree. Whereas once you become specialized, you know so much about this particular topic that you really want to be working in a particular field, and you're equipped to do that. So the 40%, pardon me, of people who are working in a job that doesn't require a college degree is opposed by 22% of people who hold a PhD or professional degree. So that halves your chances, I guess, of getting a job in the field that you want. And that meets your education levels. All right, so graduate education also opens doors in one specific, and I'm sure a very important aspect to everybody: money. If you look at this chart, we have a high school graduate earning a salary that is standardized to 1. So everybody else is compared to someone with a high school degree or diploma. So if we look at bachelor's degree, which is where most of you will be at, within 4 years or less, people with a bachelor's degree earn about 60% more than people who have a high school diploma. And once you get to master's degree level, you're earning about 20% more than you would with a bachelor's degree. If you go to the doctorate or professional, well, just doctorate. Go to the doctorate level, then you're earning about 60% more than you would with a bachelor's degree. And that averages out to about a $17,000 a year advantage. And I'll talk more about whether the cost of graduate school is worth this $17,000 extra a year. So the other perks of graduate school or graduate education are, while you're in graduate school, you get a lot of opportunities to travel. I'm here for free. And I get to see a lot of people who were here while I was here, my professors. And you get a lot of these opportunities to travel while you're in graduate school to speak about your research. You can travel to conferences, both locally and internationally. I'm looking at a conference in Spain in the summer. That should be wonderful. You also have the opportunity for personal development. Like I said, graduate school is pretty difficult. It requires a lot of self-discipline. There's nobody there to tell you to read your chapters 1 to 3. You have to motivate yourself and decide to do that because it's good for you. And so there's a lot of personal development associated with that. And once you do get a job, you get a faster opportunity to advance in whatever job you're in. I'm sorry, my voice is breaking up. I'm recovering from laryngitis. So what is graduate school like? Well, if graduate school was a relationship, you would be in a relationship with your dissertation topic. And the relationship is very complicated. 1 minute you love it, the next minute you never want to see this topic again, and you're asking yourself, what am I doing here? But I'd like to give you an example of my experience, so maybe you can have a more tangible idea of what graduate school is like. So I'm a biomedical engineering major, so I'm at the intersection of medicine and engineering. Now, inside biomedical engineering, there's the study of mechanics, so forces that interact with the body. So that's biomechanics. And then going down one level, we have the study of forces that interact with cells or are produced by cells. That's cellular biomechanics. And then my field is endothelial cell responses to flow. So I'm looking at particular cells, endothelial cells, which are the ones that line the inside of your blood vessels. And I'm looking at how they respond to blood flow. So, I'm a math major. What am I doing in biomedical engineering? Well, a lot of research that's done in biomedical engineering is based on experimental work. So you either do in vivo work where you manipulate an animal, or you do in vitro work where you're manipulating cells that are plated in a petri dish. And that work is expensive and time-consuming. I mean, there are standards that need to be followed, so you can't just pick a rat from the back of the shed and start experimenting on it. And you can't just take cells from anywhere, so this is very expensive work. So a lot of people opt for mathematical models. Thank you. This is a cheaper way to do research. It's because it's just me and my computer, and however long it takes me, and whatever cost there is for the software I'm using, to try and solve some of the problems that are going on in cells or in an animal using my computer. And there's an interaction between this experimental data and mathematical models. I have to start with some hypothesis, and I may test that in cells or in an animal. And then once I've tested it, I'm at a good place where I think, okay, this is what I think is going on, but I'm not exactly sure because there's so many variables. Let me plug this into a mathematical model and see if my hypothesis is correct. And once I have a mathematical model, that may give me some results, which I will plug back in and do some experimental data based on my findings. Also, mathematical models might point me to a new solution. I might find from my model that there's something that wasn't discovered before, and that might lead me straight to a medical application. And so we have microscopic knowledge, where you're looking at one particular aspect of a biological system. And once you do the experimental data and you have a model, then you go into macroscopic knowledge, where you have a broad idea of what's going on. So how does one decide on a research area? I was a math major, then I went to Drexel eventually. How did I decide on my particular area of research? Well, for most graduate programs, the first 2 years, whether master's or PhD, you start out by taking lots of classes, general education classes. As a biomedical engineering student, even though I have a strong math background, and I've also taken biology and chemistry courses, I also had to take biology and chemistry courses in grad school to make sure everybody is on the same level. And I took a lot of math as well. So you also attend a lot of seminars where people are presenting their research, and you learn from that research what you're interested in. People give presentations and you're like, that sounds like something I might be interested in, and you can pursue that field. And then once you're in graduate school or before, you can contact people who are doing different types of research and ask if you can join their lab and participate in the research they're doing. So my specific experience was when I left CMU, I applied to University of Missouri, among other places, and got in. And I joined their biological optics lab. So they were looking at the pupil light reflex, which is the dilation of your eye when light strikes it, in relation to autism diagnosis. Because the response is different in autistic children than in normally developing children. So I was doing mathematical modeling with that, trying to look at the response based on the muscle response, the neural response, trying to figure out if we could figure out what the difference is. I wasn't quite happy there. The research wasn't exactly what I wanted. So then I moved to Drexel, where I'm now in a cellular biomechanics lab. Here we study how cells respond to and create forces. So there's a lot of other stuff going on in the lab. We look at transport phenomena, which is diffusion and transport of molecules inside and outside the cell. We also do a lot of multi-scale modeling, which is we look at the cellular level, we look at the tissue level, and we look at the whole body level. There's in vitro work going on, so people actually test cells. And there are other people working on neural injury. So my dissertation topic, or the project I'm working on right now, is shear stress-induced nitric oxide production in the endothelium. Sounds intimidating, that was the point. So we have here a blood vessel, and the cells that line the blood vessel are called endothelial cells. This is them right there. So when blood flows, it creates a frictional force across the cells, and we call this shear stress. And one of the responses to this shear stress is that the endothelial cells will produce nitric oxide, which is, as you know, a gas. So it's a diffusible molecule that then goes to tell the smooth muscle cells to relax. So it's a vasodilator. So you have shear stress, and then that causes relaxation. So it's a feedback mechanism. So why is this work important? Well, there's lots of pathologies that lead to endothelial dysfunction. So people who have diabetes, people who have too much cholesterol in their systems, coronary artery disease, and smokers have endothelial cells that don't quite work the way they are supposed to. And this is marked by nitric oxide production that is not as high as it should be. And this in turn leads to other problems: atherosclerosis, heart attack, and high blood pressure. So we need to figure out what's going on in the endothelial cells that goes wrong in endothelial dysfunction, so that we can possibly identify drug markers to fix the problem. So this, from experiments that have been done throughout the literature, people who are much older than I am, and have been in this business longer than I am, have established that for an endothelial cell, where this is the cell membrane, and this is the endoplasmic reticulum membrane, there are these cave-like structures called caveoli. And there's a whole bunch of proteins that are embedded in this structure. And all of them seem to be important in this response. So it's interesting that they're grouped together, because that means that there's kind of compartmentalization. Everything can happen in a specific location, and things don't have to diffuse far to do that. So I won't go into the nitty-gritties of the pathway. Just know that there's flow, and then there's a series of reactions that occur inside the cell. To basically summarize or simplify that signaling cascade, we have shear stress, which is the force of blood flow across the cells, and that causes ATP production. So I have shear stress, my cells produce ATP, and then there's autocrine signaling. So once a cell feels that there's a high concentration of ATP around it, it produces more ATP. And that tells the cell to release calcium from the endoplasmic reticulum. And that tells the cell membrane to open some channels and let more calcium in. So now I have a whole bunch of calcium inside the cell. And because the calcium concentration is high, I have this enzyme which produces nitric oxide, my diffusible vasodilator, and it increases in activity and starts producing nitric oxide. So some experimental work that has been done suggests that this is important, but we're not sure. It suggests that this calcium entry from outside the cell is important, but we're not sure. It suggests that protein called PKC is important, but we're not sure. So I have to find a way to use mathematical modeling, which is, like I said, cheaper, faster, and more efficient than doing more experimental work to verify this, and try to figure out, okay, is this true? Is this what's really going on? So I'm asking the questions: does this relationship explain what's going on experimentally? And also, if I put this into a mathematical model, can I play with the mathematical model and cause nitric oxide production to decrease? So can I reproduce on my computer screen endothelial dysfunction? And if I can, that means whatever I've fidgeted with may be what's going wrong or what's impaired inside the cells of people who have endothelial dysfunction. And once I have a working model, I can also ask the question, what predictions can I make? I'm going to plug some other stuff into my working model and manipulate it and see if I can tell anything else that might be happening in the cell, something that people haven't even thought about doing in their research. All right, so this is my mathematical model, a whole bunch of equations. I'm not going to explain any of them. I just want you to know that this is it. So this is where my math comes in. I have some differential equations. If you can see, dIP3 dt. I have some Michaelis-Menten relationships. Those of you who do chemistry, you know what that is. And obviously, I have to have some understanding of biology if I'm going to put these together. And where the computer science part comes in is there's no way, those of you who've taken differential equations, there's no way I'm going to write this set of equations down and solve it with pen and paper. So I'm going to plug it into a computer, MATLAB in my case, and let the computer solve the equations numerically. So what do these equations represent? I'm basically looking at a value of how much of each molecule is in the cell. So I say I have X amount of calcium, and it changes depending on how much ATP is produced. So that's basically what that is. And this is much easier than my plating cells, incubating them for a week, waiting for them to look like an endothelium, testing them, which takes about a week, looking for results, discovering that I didn't put the right concentration, and then having to start all over again. So what kind of results do I get from this mathematical model? So we have here some experimental work done by another group where cells were plated and were put in a flow chamber, which is basically an apparatus that has cell culture medium flowing over it. And I expose these cells to about 1 pascal of shear stress for 3 seconds, and I want to see how they respond in terms of how much ATP is produced and how much calcium is produced. So this outline is the shape of one single endothelial cell, and I've marked my ATP so that a high concentration comes out as red and a low concentration comes out as blue. And the same thing for my calcium. So the surface of an endothelial cell, I see there's one specific location where after 3 seconds I have a very high concentration of ATP, and the same location I have a very high concentration of calcium. That goes back to those cave-like structures, the caveoli I mentioned. They're not everywhere in the cell, they're some places in the cell. So now I've established that there can be specific locations, these caveoli, where all these proteins are together and things seem to happen together in the same locations. So if I put that into a graph, I see that for a 3-second stimulus, I start out at a low level of ATP, which is what this represents. It increases as soon as the flow begins, and it decreases before the end of the stimulus. At the same place I measure calcium, I see there's a bit of a lag, about a 1-second lag, before calcium starts increasing. That's coming from the calcium that's released from inside the endoplasmic reticulum, and the calcium that's coming into the cell from outside. And it goes back and decreases. So now I want to see if my model can reproduce this behavior. So the equations I use are phenomenological. I didn't account for every single behavior, otherwise my equations would be much hairier than what I have here, but I've simplified it to where I'm comfortable that I think I can represent this behavior. And this is what I get. I pretty much get an increase in ATP and then an increase in calcium. This is not for the same run of the cell, so this process is stochastic. Not every single cell is going to behave in the exact same way. Cells have different numbers of receptors, they don't feel well that day, they just respond differently. But I can see that I get the same type of response, so my model is representing my biological phenomena correctly. So now I can start manipulating the model. So, as you may have seen from my hairy set of equations, there are a lot of constants or parameters. And because this is a simplified model, there's nowhere I'm going to look up in a textbook and say, what's the value for A sub 4? And I have nowhere to find that. So I have to figure that out for myself. I know this is what the relationship looks like, but I need to figure out what parameters go with that relationship. So I use my computer to tell the computer what experimental work looks like, that's the red dots, and then I tell it this is what my equation looks like, but I don't know what the correct parameters are. Can you look at this, can you look at this experimental result and look at the relationship I have and fit this model to the experimental work I have? And can you tell me what parameters would work? I know that the parameters are going to be in this certain range. If it was temperature in the body, I know it's going to be a little less than 37 or a little above 37. So that's my range that I'm working with. In the same way with my parameters over here, I have certain ranges that I know the cell is never going to have a parameter that's less than this or higher than this because it's physically impossible. So, but can you tell me exactly what it is? So I plug it in, and it spews out some parameters, and it plots the graph that goes along with the parameters. So that's a pretty good fit. I've put some equations down on paper, and it basically represents what's going on inside my cells, so that's pretty good. So now I'm going to try and manipulate my model. I'm going to say, all right, the cell produces ATP when shear stress is present, and then that ATP causes more ATP to be produced. If my receptors that tell the cell that there's lots of ATP outside are broken, the cell doesn't know that it's producing lots of ATP. What does that look like? So in my mathematical model, I set some value equal to 0, and I get this graph where the cells are not producing autocrine ATP. The cell is just producing this initial amount of ATP. And what that tells me is that cells that don't have these receptors that they need to sense ATP production will still have a response, but it'll be very low. Maybe that propagates downstream to nitric oxide. Maybe this is where things are broken in people who have endothelial dysfunction. On the other hand, it also tells me that because I have a fairly flat surface on the top of this response, it tells me that maybe the cell has a certain response. I have a certain level of shear stress, and that produces the maximum value for this. If I was to increase the amount of shear stress, if I was to make the blood flow faster or harder, then I would have a plateau that's higher. So that tells me that the initial release of ATP is a set point that tells the cell how much shear stress it's under. So that's also something important, that because we don't know what in the cell is responding to shear stress, will guide experiments down the line. So I'm not going to go into any more results. I'm about to run up. I'm going to tell you what I'm planning to do when I graduate, if they let me graduate. So hopefully, or not hopefully, maybe, I'll do a postdoctoral position, which is where once you have your PhD, they're not going to let you into your lab to start doing your own thing or supervising students. They're not going to let you teach yet, but they'll let you work in a lab that's headed by someone else, where you can do your own research independently. And once you've shown that you can do that, then they're willing to give you a job or a teaching position. I'm pretty young and I don't have patience, so I don't think I have the patience to teach. As a graduate student, I teach students, and I've lost a lot of hair. So I think my plan is going to be to work in research either in industry for companies that are doing biomedical research, or to do consulting, which is to work for a company that works for companies that are doing biomedical research. Eventually, when I have gray hair and a lot more patience, maybe I'll go back into academia. I'll be a lot more mellow then. So, you've heard what graduate school is like. I've suggested to you that there's a lot of money involved. There are a lot of motivations for going to graduate schools, and I'm going to suggest to you that some of them are good and some of them are not so good. So people go to graduate school for necessity. You want to be called doctor when you grow up, so you need a graduate education to do that. Or whatever it is what you want to do when you grow up requires a graduate degree. That sounds like a good reason that you're going along the path to meet your goals. There's also freedom. If you don't want to work for somebody else and you don't want to call somebody else sir, you may want to go to graduate school, because that gives you a freedom to be more or less your own man. When bureaucracy comes into it, that's not quite true, but it's nice to think it is. A lot of people, like I put up the statistics about money, a lot of people will go to graduate school because they want to have a better salary. I'm going to propose that that's a good and a bad thing. It's a good thing. You want to live the lifestyle you want to live. But if you're going to graduate school just for money, it might not be such a good idea. This is a graph of different professions in academia and their salary. While you're a graduate student, you may have the opportunity to work and you'll be paid a measly $17,000 a year. You may not be paid, so your salary is basically zero and you're mooching off your parents. Once you become a professor, if you decide to go into academia, then you go up the ladder and start earning that 75%, that's $17,000 more a year. And if you stay in academia, you make your way up that graph. But if that's your only motivation, then that may not be such a good idea, because while you're here for 5, 6, 7, 10 years, life is gonna suck. So if you're in it for the money, be a football coach, because you're going to make $1 million a year. So if you take nothing else away from this, this is my advice to you. So other motivations for going to graduate school. If you like the sound of doctor at the end of your name, that may not be such a good reason. A lot of people do like the sound of doctor behind their name. If you call the person sitting next to you doctor, whatever the name is, that's going to put a big grin on their face. But it's going to take a lot of work to get there. If you're avoiding personal, financial, and family obligations, it's not such a good idea. Some people go to graduate school to wait out the economy, but heaven knows what's going to go on with the economy. You might come out with a graduate degree and not be as employable as you thought you were. If you don't want to find a job, you're going to have to find a job eventually. This is a reality I'm coming to grips with. that's not a good reason to go to graduate school. And if you don't know what to do with your life, I'm kind of ambivalent about this one because I don't know what I want to do with my life. I don't have a specific job that I have in mind. I have a general idea. So I know that I want to do something that requires a graduate degree. And in the general area, I know what that is, but I don't know specifically. It's not like I have a job lined up for me. But I think that's okay. But if you graduate with a general studies degree and you're not sure what you want to be when you grow up, that's no reason to go to graduate school. So a question that a lot of people ask is, yes, we've seen that we can make a lot of money. It's going to be wonderfully rewarding when you go to graduate school. But how am I going to pay for this thing? Graduate school tuition is pretty steep, anywhere from $30,000 a year going up. There are a lot of jobs that will pay for you to get a master's degree, depending on where you go to work. If you work for a lot of these big corporations, in biomed it's going to be big pharma. They will pay for you to go to graduate school on a part-time basis. And a lot of graduate programs have classes at night, so that's not a problem. You can also go for free if you find a lab that's willing to have you as a research assistant. That's where that $17,000 comes in. They'll pay you a stipend and waive your tuition fees. So you can, well, not quite for free, I guess you're getting paid to do research. You can also be a teaching assistant, and there are no special qualifications required. Once you have a bachelor's degree, they're assuming you know at least something about the topic you majored in. So they'll let you teach undergrads, freshmen mostly. That's where the hair pulling comes in. So my advice to you is, if you want to do either of these two, or if you want to go to graduate school full stop, definitely get involved in research at CMU. I know a lot of people think there's not much research going on here, but I know there are some professors who are doing projects. I don't know specifically what those are. And I also know that there are courses you can take here. I was in the honors program and I got to do an independent project. I also did another project for, I can't remember what, but there are lots of projects you can do alone with the help of your professors, which constitute as research. And that is very good because getting a research assistantship position is competitive, because nobody's going to give you $17,000 a year, even though it's not a lot, to mill around if you don't quite know what you're doing. So that's it for my talk. I'd like to thank the following people for facilitating this event. I kind of invited myself, but Nicolette made it possible for me to be here. Thank you for the Science Division for having me, and for all of you for attending. The Drexel University Graduate Studies Office gave me a travel award to be here to tell you about graduate school. And of course, my PI and people in my lab, and the greater research group that I'm in. Thank you very much.